nobody bothered to look into this association. Okay, it was in 1950s. You can see the gap, 1884 to 1950s. Practically nothing has been done in this particular area of science. And in 1950, a student from University of London was asked to select her thesis problem. So she was asked to select, and we know here the teachers give the thesis problem to the students, but in Western countries, the student has to select the thesis problem. So when she was asked to select the thesis problem, she saw that the German botanist has described mycorrhizal association, but nothing has been done. So she wanted to work on this for her thesis. And she worked on mycorrhizal association in apples. Of course, in UK, apple is a very important crop. So she chose to work on it. So after submitting her thesis, and that was a classical work on mycorrhizal association. After that, she joined experimental research station in Rothamsted. Rothamsted Experimental Station is considered to be the mecca for agricultural scientists, so the topmost agricultural research institute. And she joined there and continued her work on mycorrhizal association. And when she started working, another extremely good thing she did, that is, she invited people from all over the world, whoever is interested in mycorrhizal research, to her laboratory with the fellowship. Let me underline that, with the fellowship, so that they can learn about mycorrhizal association and the research associated with it, so that they go back to their own country, and so that the research on mycorrhiza spreads to different places. That is how now we have the mycorrhizal research all over the world. And that happened during her period. And now we are in 2021 beginning. Now we have four main types of mycorrhizal associations. Ectomycorrhizal associations, which are commonly found in temperate forestry species. Vesicular arbuscular mycorrhizal association. Now it is called as arbuscular mycorrhizal association, which is found in most of the agriculturally important crop plants. Then ericoid mycorrhizal association is associated with plants belonging to Ericaceae, like blueberries, cranberries, azaleas, rhododendrons. Again, temperate. In our country, it occurs only in the Himalayan region. And fourth category is Arctic mycorrhizal association. As the name itself indicates, it's very specific, occurring in as orchids. But I am going to concentrate only on the second category, and where a lot of work has been done, because that's the one which is very important in agriculture. Okay, now we see this the cross section of a non-mycorrhizal root. So we see the epidermis, and we see the endodermis, which is in the middle, and we have the cortex. Okay, this is the cross section. And let us see here a root which has been colonized by the arbuscular mycorrhizal fungus. So the fungus lives in soil, and in the soil, it produces a very large spore called as extramaticular chlamydospore. It's quite big. It can go up to 450 micrometers, quite big, such spores. And the hypha penetrates the root through the epidermis and then ramifies in the cortex region. It never goes beyond the endodermis, never penetrates the endodermis. So it is restricted to the cortex region. And in the cortex region, it produces large globular spores called as vesicles and highly branched hyphal structures called as arbuscules. That is how they were called as vesicular arbuscular mycorrhizal fungi. Later on, it was found some genera of ampfungi do not produce vesicles, and therefore they removed the word vesicular, whereas all of them produce arbuscules. That's why now, if you see the literature after about 1990 or even little later, you will see only arbuscular. If you see the earlier literature, you will see them referred to as vesicular arbuscular mycorrhizal fungi. Okay, Now, this is ubiquitous. This association is ubiquitous. What I mean by is they are present everywhere. Everywhere, if you go to the US or you come to India or you go to Nigeria, take the soil or take the plant root and see whether it has the mycorrhizal association. It definitely has, because 90% of the crop plants grown have this particular association. So even in your campus, in the college, we go out, pull out any plant, and then come bring it to the lab, stain it with a particular stain called as trypan blue. I can say that you will definitely see these beautiful vesicles which are inside. So when we say the root, it is always associated with the fungus. And these are the vesicles. And the ones above, these are the arbuscules, which are highly branched hyphal structures. The same thing is magnified here, and it is dichotomously branched usually, and they are short-lived, just 4 to 5 days, because they undergo digestion and they just disappear. So it's only 4 to 5 days. And these are said to be the organs through which the nutrients from the soil is transferred to the plants. Okay, or vascles is the place where the transfer of nutrients take place. All these fungi, fungi, belong to one phylum, Glomeromycota, which has 3 classes, 5 orders, and 14 families, and 26 genera, and 230 species described so far. Of course, this taxonomy varies with the different taxonomies. Some others say that it has more number of families and more species and so on. But this is what is usually accepted. So I said there are 26 genera of amphifungi and 230 species. Though there are 26 genera, the 4 genera are very common in most of the soils. Okay, so A here is glomus, and B is Gygospora, and C is Achilospora, and D is Entrophospora. These 4 genera are the ones which are very common in almost all soils throughout the world. And of these 4, if you ask the question, which is the most common? The answer is A, that is Glomus. Glomus is the most common genus occurring in most of the soils, including Indian soils. So if you just again go out to your college campus or university campus, take the soil, bring it and do the analysis and see under the microscope, I am 100% sure that you will get glomus. That is so common, including Bangalore soils. And these fungi are obligate symbionts. What do I mean by obligate symbionts? So the thing is, we know fungi. How do we grow fungi in the laboratory? We use potato dextrose agar or Richards medium or Zipex agar for growing this. But this fungus, arbuscular mycorrhizal fungus, cannot be grown on a laboratory medium. So it needs a living host root for multiplication and growth. So here, this is how we maintain the arbuscular mycorrhizal fungi in my institute. You can see we maintain them along with the host, that is usually the grasses, and maintain it in a glass house. Okay, so each row is one species of amphifungus. So this is how globally in the culture collections the amphifungi are maintained, even in all over the world. And here again, I want to go back to the history and then say how we started mycorrhizal research in Bangalore, or I can say how we started it in India. Okay, I had a Fulbright fellowship to go and spend 1 year and in the University of California. The assignment was to transfer the NIF gene, that is the nitrogen fixing gene, from rhizobium to a saprophytic bacterium, Yervinia herbicola. So that was the purpose with which I landed in University of California, Davis campus. And the next lab was working on mycorrhizal fungi. So the initial first few days, I used to peep into the next lab. And somehow what the mycorrhizal lab was doing interested me more than what I was assigned to do. So I went to the Professor Mortimer Starr, who was the editor-in-chief of the annual review of microbiology. And so I told him, can I shift to the next lab? Then he said, you come on a Fulbright fellowship and the office is in Washington, D.C. You talk to the Fulbright office people, and if they approve, and then I have no objection. So I called the Fulbright office in Washington, D.C., and the lady took the phone and she said, if the professor, if both the professors have no objection, we have no objection. So that is how I've shifted to the next lab, So that is how I got interest in mycorrhizal research. And there is always a common saying, the neighbor's food always tastes better. So that is, it happened with me also. So that is how I started shifting to the next lab and started working on mycorrhiza. And at the end of the 1 year period, it was December 31st, I still remember it very vividly. And about 2 months before my fellowship was coming to an end, the professor asked me, we can take you on a contract basis or for 5 more years in the same laboratory. Would you like to continue in my lab for another 5 more years? And also he said, that can continue further also. Okay. Then I thought, I talked to my people at home in Bangalore. I talked to my mother and I talked to my wife. Both of them said, come back. And more than that, I talked to the Indian people working in the University of California, and they said, we are sticking on here because we did not get a fairly good job in India, but you are already an associate professor in a government university and you should go back. So I decided to come back. Many people said that I am a fool doing the wrong thing, but no regrets. What I have done was the best. So when I came back to the University of Agricultural Sciences, which deputed me to US, and we were not sure whether mycorrhizal fungi occur in India. That is how we have to start from a scratch. And so I asked my students, please go out, bring the plants from the campus, let us stay in and see whether they are mycorrhizal. And these are the 3 slides which I have put here, root samples done then many years back, decades back. Okay, we found that they were mycorrhizal. So we were saying very definitely, we know that our plants are all colonized by mycorrhizal fungi. Then we brought the soil samples, did the wet sieving, and then looked for the spores under the microscope. Plenty of mycorrhizal spores were there. So that means our soils have the fungus. Okay. Then when coming back from US, the professor asked me, what do you want from here? So I said, the best mycorrhizal fungus you have. So they used to give one mycorrhizal fungus, Glomus fasciculatum, to the farming community. Usually the people growing oranges and avocados, the 2 main crops in California. So they were giving this fungus. He said he was very happy to give me that particular fungus. So I brought the fungus and wanted to see how it works in Indian soils. So this is the first experiment we did in Bangalore. And we autoclaved the soil and inoculated with Glomus fasciculatum. This is the type of difference we could get in Bengal gram. We were really surprised to see this difference. And then we said, our soils also have native mycorrhizal fungi. So, okay, we did a mistake by using autoclaved soil. We should use a natural soil. So the next few experiments, we used natural soil and then saw the difference to inoculation with the fungus, which I got from US. So this is the difference, which very clearly showed, not only this is in case of groundnut, this is in case of cardamom, this is in case of neem. I'm just showing 3 plants, one important in agriculture, one important in horticulture, one important in forestry. So this much of difference you could see to inoculation with Glomus fasciculatum, which showed that that is a better fungus, more efficient fungus than the native fungi what we have here. So which then the question is, how do these mycorrhizal fungi improve plant growth? Is the next question. So I just take one example here. As cotton, I is inoculated, U is uninoculated. You can see the difference in dry weight of the plant, difference in the nutrient uptake, phosphorus, zinc, manganese, which showed significant increase in uptake of nutrients. So we could conclude it is because of the uptake of nutrients. The next question, what type of nutrients? Again, we have to go back to little soil science and say plants in the soil, the nutrients are two types. Some nutrients move freely in soil solution. So these we in soil scientists call them that nutrients moving through mass flow. Nitrogen, potassium are examples. They move very freely in soil solution. Some nutrients do not move freely in soil solution. They are referred to as immobile nutrients by common people, but soil scientists will call them as diffusion limited nutrients. So these nutrients do not move freely in soil solution. Example is phosphorus, zinc, copper, etc. Okay. So now what happens? Let us take an example. And this is a plant root. And we all of us know that plants take nutrients from the soil through root hairs. So these root hairs absorb the nutrients. And we also know plants take phosphorus in the form of H2PO4 ions. So all the available form of phosphorus, H2PO4 ions, are taken by the root hairs. They have taken away all the phosphorus. Okay, there is no more phosphorus left in the root hair zone. So that is what we call as P depletion zone, because they have taken away all the phosphorus. Same thing applies to zinc and copper, which are less mobile or immobile in soil solution. So now what happens? There is no more phosphorus for the plant roots. So here the mycorrhizal fungi comes into picture and plays a very, very important role. So we know that the hypha of AM fungi, they travel a much longer distance compared to root hairs, several centimeters, and they take the phosphorus and then pump it directly into the host, thereby short circuiting the supply of nutrients. So this is where the mycorrhizal fungi play the most important role in the uptake of diffusion limited or immobile nutrients like phosphorus, zinc, copper, etc. And in addition to that, in what other way they help plant growth? Being a microbiology lab, we were interested in studying the interaction between the mycorrhizal fungus and the nitrogen fixing bacterium, Rhizobium japonicum, in soybean. And so we had in the campus, we had a project on soybean by the agronomy group. So this was a field experiment. And again, I would like to tell, most of the mycorrhizal workers have done all the experiments through pot cultures. So very rarely they went into the field. So this was one of the very early field experiment that was done to study the interaction between AM fungus and the rhizobium. And you can see here with only inoculation with the mycorrhizal fungus, the grain yield was this much per plot, 56. With rhizobium inoculation alone, it was 84 grams per plot. But when both with the co-inoculation, when both were high, then there was a significant increase in grain yield, which very, very clear. The same thing was true for nodule number, mycorrhizal colonization, dry weight, and so on. So which very, very clearly showed there is a synergistic interaction between the two symbionts. And a dual inoculation is much better in increasing the growth, nutrition, and yield of plant compared to inoculation with a single organism. it was very clear from this particular experiment. We did a similar experiment with a free-living nitrogen-fixing bacterium, Mustobacter, with the mycorrhizal fungus, which again showed the synergistic interaction between the mycorrhizal fungus and the free-living nitrogen-fixing bacterium. And coming to the next group of another organism which is present in soil, which is agriculturally important, the phosphate solubilizing organism. Yesterday I mentioned about the importance of phosphate solubilizing bacteria and fungi. And here I have to say something, that there was some difference of opinion amongst the mycorrhiza workers. Some of the mycorrhiza workers felt that the mycorrhizal fungi can solubilize unavailable form of phosphorus and release the H2PO4 ions which the plant roots can take. Another 50% did not believe this. So there was a controversy between the mycorrhizal fungus. So we took up this study using P32 labeled tricalcium phosphate, and we distinctly proved that mycorrhizal fungi do not solubilize unavailable form of P. They only help in the uptake of available P from the soil. So here, that is why we studied the interaction between the phosphate solubilizing organism, which can solubilize unavailable form of P, and the mycorrhizal fungi, which helps in the uptake of soluble P. So again, we could see there was a synergistic interaction between the two organisms. So how does this work? Suppose the unavailable form of P, let us say rock phosphate. A farmer has used rock phosphate in the soil. So the phosphate solubilizing organisms act on this rock phosphate, producing organic acids, and they release the phosphate ion. And this phosphate ion is taken by the mycorrhizal fungus. So that is how there is a synergistic interaction between the phosphate solubilizing organism and the mycorrhizal fungus. Then another aspect which we studied was whether they have any role in the biocontrol of plant pathogens. Again, this was done in my first PhD student. And so here you can see he worked on groundnut, and this is a controlled healthy groundnut plant. And this is the plant diseased with the pathogen Sclerotium ralsei, which causes soft rot or wilt of groundnut. You can see how the plant looks like, the diseased plants look like. Here, the third one, you can see the plants inoculated with the pathogen plus the mycorrhizal fungus. Okay, so you can see the difference between the second and the third, which very clearly shows the mycorrhizal fungus has helped the plant to alleviate the disease caused by the pathogen. So it looks much better when it is inoculated with the mycorrhizal fungus, though the pathogen has infected, but it has less damage, it has caused less damage. So we were wondering how the mycorrhizal fungus protects the plants against the pathogen. We went into the details and found that the roots of mycorrhizal plants has higher concentration of OD phenols, orthodihydroxyphenols, which prevented the germination of the pathogen, the sclerotium of the pathogen, Sclerotium ralsei. That is how it has protected the plants against the pathogen. Again, another pathogen, which is very common, causing disease in and around Bangalore. Yesterday also I mentioned about it. That is the root knot nematode. It's a very serious problem in and around Bangalore and in Karnataka also, in other parts, that root knot nematode damage, and especially on solanaceous crops. This is tomato root system, and you can see here it has been infected with the root knot nematode. You can see the big galls. It's called as tertiary galls, and so which in turn brings down the productivity of the crop, of the plant. Here we have used the same number of larvae of the nematode plus the mycorrhizal inoculation. So here what happened, it produced very small galls called as primary galls, okay, and which did not develop bigger as secondary or tertiary galls. So we were really breaking our head how to explain this. Okay. In mycorrhizal plants, there were only primary galls, but which did not develop into tertiary galls. So we discussed with our nematology group of scientists, went through the literature, and finally we could get an answer. That is, the in inoculated plants, when the nematode has entered the mycorrhizal root, and that is how it is producing the primary gall. That means the mycorrhiza is not preventing the entry of the larva into the root, and that's how we have the primary gall. But what is doing the mycorrhiza doing? It is preventing the larva to develop into adult. If it is preventing, that is how we are not getting secondary and tertiary galls. And what is it that prevents the development of the larva, nematode larva into adult? Again, going through the literature, discussing with our nematology group, we could see the sulfur containing amino acids prevent the development of larva into adult. So we analyze for the sulfur containing amino acids in the roots of mycorrhizal roots, and we could see it has a high concentration of sulfur containing amino acids. Now we can answer that why it is preventing the development of nematode galls. So what now I have just given in a nutshell, what are the various mechanisms of improved plant growth because of mycorrhizal inoculation? Increased nutrient uptake. Naturally, this is again mainly of the less mobile nutrients. Then synergistic interaction between other soil organisms, especially the nitrogen fixers, P solubilizers, phosphate PGPRs, and so on. And protection against the soil-borne plant pathogens. And better drought tolerance. They also help in the uptake of water. And a drought situation, now water is becoming a rare commodity, and many farmers are facing this problem of water stress. So we had one project funded by ICR which just ended very recently, and we have shown very, very clearly that mycorrhizal fungi protects the plants when they are given only 50% field capacity, that is the of the requirement of water. Okay, so this has been done under field situation and using soybean as the crop, and this is a very good results we got, and showing that the mycorrhizal fungi helps to overcome water stress. And it also produces growth promoting substances like indolestic acid, gibberellins, cytokinins. So it is a cumulative effect of all these which makes the plants grow better. Here, this is an interesting slide. Here, one of the students did this experiment. You can see that last one is the control. And all these are the plant is Lucina leucocephala, a forestry species. And we can see that they have inoculated with different mycorrhizal fungi, different TAM fungi. And looking at this, what can you say? The last one is the best mycorrhizal fungus for inoculating Lucina leucocephala. Here I have to say something. All the mycorrhiza workers earlier, including Dr. Barbara Mose from England, whom we call as mother of mycorrhiza research, and Professor Gardiman, who started mycorrhiza research in US, in all the reviews till 1980s or 1985 also, I can go one step further, and even 1990s, they said the mycorrhizal fungi are not host specific. Okay, it is true. Even today, I can agree with that. They are not host specific. What do we mean by not host specific? You isolate the mycorrhizal fungus from ragi plant and try to inoculate tomato, it will colonize tomato. Take it from tomato and try to put it on banana, it will colonize banana. Put it on a forestry neem tree, it will colonize neem. That is why we say that they are not host specific. But from this experiment, used a new hypothesis. We said there is host preference in mycorrhizal fungi. Though they are not host specific, they have a host preference. That means for a particular host, there is always the particular fungal partner which produces the maximum symbiotic response. So what does this mean? And it means that for if you are interested in a particular crop, let us take tomato as an example. If you are interested in tomato as a crop, you can screen different mycorrhizal fungi and select the best mycorrhizal fungus for inoculating tomato. This is what we meant by host preference. But there was so much of opposition. People from UK and USA did not accept this host preference hypothesis. So we have to show it in many other crops. This is in cashew, and we said this is the best fungus. Last one is the best fungus for inoculating cashew. Again, this is the citrus rootstock, and we said the one here, this is the best fungus for inoculating rootstock. With all these, they were convinced, I hope so, they were convinced, but they're not fully convinced. They wanted to get convinced by giving us a big project. Okay, the United States Department of Agriculture gave me a very big project and said, if your hypothesis, if a host preference is correct, we are giving you this project. And they said, you have to use a forestry species. And then this project is for 5 years. At the end of 5th year, we want one particular fungus which can be used by the forest nurserymen to inoculate the forestry species. So that is how the project was given. I was very happy. The university was very happy. Lot of money came in US dollars, so which could help many students to work. And before going to that, I would like just give one particular thing which we also introduced, that is called to determining to what extent a plant is dependent on mycorrhizal fungus, and which we call as relative mycorrhizal dependency. And so this is given by a simple formula: the dry weight of the plant grown in unsterile soil minus the dry weight of a plant grown in sterile soil divided by dry weight of the plant in unsterile soil into 100. So this gives the percentage. So if a plant is highly dependent on mycorrhiza, it will have a high percentage of RMD. So based on this, we have classified the plants grown in Karnataka state into 4 categories. Those which are highly dependent on mycorrhiza, which are moderately dependent on mycorrhiza, less dependent on mycorrhiza, and not dependent on mycorrhiza. So what we suggested is to work on the plants which are highly dependent on mycorrhiza to begin with. Okay. And Lucina leucocephala is a plant highly dependent on mycorrhiza. So we chose this particular plant to work on the project given by the United States government. So we screened various mycorrhizal fungi and selected the best. And again, there are 4 different cultivars of Lucina leucocephala grown in Karnataka state. Okay, K8, K28, and another 2 more. So we selected a fungus which will work on all the 4 commonly cultivars that are grown in the state. The reason is, suppose one forest nurseryman or a farmer asks us the best culture, and he may be growing K8, and another farmer may be growing K28. We cannot give different cultures to different farmers. So we wanted to give one culture which is the best for Lucina leucocephala. So that is how we selected the best one which works on all the 4 cultivars which are grown in Karnataka state. And this shows the difference in the root system of the 4 cultivars. You can see the difference uninoculated versus inoculated. See the significant difference in the root system. So at the end of the 4th year, we gave this particular culture. US government was happy with the result, and Indian people, foresters, were very happy that we have a culture which can be used for inoculating this plant, and which was introduced to India as a wonder plant. And so when this work, okay, before that, and in agricultural universities, we have a Farmers' Day, which we call as Krishi Mela. Okay. Then all the professors and scientists working in the university, we have to face the farmers. Okay, I tell you, young people, here, it is much easier to face scientists and students. It is much more difficult to face farmers. They ask such intelligent questions. In fact, to be honest with you, I have developed some research projects based on the questions raised by the farmers, because they really ask such interesting questions. So, I was presenting the work on Lucina leucocephala to the farming community, and one farmer stood up and asked me the question, which is the best fungus for inoculating Lucina leucocephala? I told him it is Glomus mossae. And second question he asked, which I never expected, which is the next best fungus for inoculating Lucina leucocephala? I told him it is Gygospora margarita. He came with the third question. The third question was, suppose I mix both these fungi and inoculate, what will happen? I never had an answer. So I told him, we have not done that work. Please come next year. I will do this and tell you what will happen. So we did this, mixing these two organisms, and with compared it with the single inoculations. And we could see that mixing these two organisms has no disadvantage. And so we can easily mix these two organisms. Maybe it may be advantageous or in one soil which is having, which may not work very well with the first fungus, it may work with the second fungus. So there's nothing wrong in mixing the two best fungi and inoculating it. And so at this time, it so happened that the Director General of ICFRE, that is the Indian Council of Forestry Research and Education, and that is the top institution for forestry in India, from Dehradun, the office is in Dehradun, and this Director General happened to visit the Bangalore Agricultural University. And the Vice Chancellor of the university usually brings any visitor coming to the university to two departments, plant physiology and microbiology. So as usual, he brought the Director General to microbiology, and he told me, you just show your experiments in the glass house and in the field. I took him to the glass house and showed the difference in Lucina leucocephala inoculated with the Glomus mossae. And he saw that and he said, can your fungus bring this much difference in plant growth? Then he said, sir, you are seeing that is what we have done, nothing else. He was so surprised and he said, if I give you a project, can you develop such inoculants for various forestry species? Again, a big project came from ICFRE. So we were very lucky. I could help many students with projects. And so we have developed the best fungus for inoculating various forestry species. I'm just showing you one. This is teak, Tectona grandis. You know how valuable timber it is, Tectona, the teak wood. And so you must be, all the furnitures in the house, in the posh houses are made up of teak wood. So this is the type of difference we are getting when we inoculate with the selected mycorrhizal fungus. We have to screen and select the best mycorrhizal fungus, and we have done that for various forestry species. And this is the list I am giving you here. We have done and selected the best mycorrhizal fungus for inoculating various forestry species like Acacia nilotica, Acacia auriculaformis, Albizia lebec, Azadi recta indica, Casuarina equisetifolia, Tamarindus indica, and all these have been handed over to the forest department for their use for raising the seedlings in the nursery. And when this work was going on, the horticulture people came to us, the horticulture department. We want such best fungus for inoculating various horticultural plants. Again, a project came and we could develop the best mycorrhizal fungus for inoculating various horticultural plants like tomato, capsicum, chili, coffee, cardamom, including the ornamental plants like marigold, flowering plants, chrysanthemum, and also the medicinal plant like rosemary and so on. So this was again, following this, the National Medicinal Plants Board came to us and said, again, we want the best mycorrhizal fungus for inoculating various aromatic and medicinal plants. Again, another project came. So I was very lucky in getting project after project. So we have developed the best mycorrhizal fungus for inoculating various medicinal plants and aromatic plants. And okay, after developing the best mycorrhizal fungus, what next is the question. How it is be of practical importance, how it will be going to useful to the farming community, the stakeholder. All this we are doing to help the farming community. And so we did this simple experiment, which we call as phosphate response curve. So here we have the increasing x-axis, we have the increasing phosphorus fertilizer level concentration. And the y-axis, we have the plant biomass. And we have the non-mycorrhizal plant and we have the mycorrhizal plant. Okay, let us take the non-mycorrhizal plant. To the non-mycorrhizal plant, to produce this amount of biomass, it has reached the maximum, it produces this biomass. How much of phosphatic fertilizer is required? Let us say 70 kg T. And for the mycorrhizal plant, to produce the same amount of biomass, how much of phosphatic fertilizer is required? It is much less than 70, let us say 50. So that means the mycorrhizal plants, through mycorrhizal inoculation, we can save 20 kg of phosphorus. This is what is very important. Why? Because the phosphatic fertilizer is not only expensive. For the manufacture of phosphatic fertilizer in India, we are importing the raw material from Morocco till today. So again, lot of foreign exchange goes for the importing the raw material for the manufacture of phosphatic fertilizer. So Government of India is very much interested in this particular aspect of reducing the foreign exchange. So that is how importance is given to this particular kind of research. So we have done this at field levels. I'm just with several crop plants. I'm just giving one chili. And the recommended level of phosphatic fertilizer for chili crop is 75 kg P per hectare. And 50% of 75 is 37.5. Okay. So what we have done for the uninoculated chili crop, and we have given the recommended level of phosphatic fertilizer. With the mycorrhizal inoculation with different species of mycorrhizal fungi, we have given 50% of phosphatic fertilizer with the mycorrhizal inoculation and compared it with the 100% phosphatic fertilizer given to the uninoculated plants. You can see this is the yield, 1.81 kg, 1.81, and here, and this is the kg per plot. And here with the 50% with the mycorrhizal inoculation, it is 2.14, which very, very clearly brings out that 50% of the recommended phosphatic fertilizer to a crop can be brought down through inoculation with an efficient mycorrhizal fungus. Okay, this we have done with several crops. So this is one which is very, very important, which attracted the end user and also the funding agencies. And here, I just want to say everything is high-tech nowadays. So agriculture is also growing high-tech. So the farmers in and around Bangalore never raise their seedlings nowadays. They just go to a nursery man, buy the seedlings and plant in their field. So the nursery men raise the seedlings and sell it to the farmers. So that is the situation now. So here, this is a nursery man raising the asparagus seedlings here in a root trainer. Okay. So here, this is the close-up, how it looks like. So here, the technology, mycorrhizal inoculation technology becomes so simple. They can inoculate, mix the mycorrhizal inoculum with the substrate, and then raise the seedlings. They all become mycorrhizal. When the farmer goes and buys it, and he takes the seedling and plants it in the field. The mycorrhiza goes along with it. So he gets a very good crop, mycorrhizal inoculated crop. So the simple, very simple technology. At this time, again, the horticulture people had another problem and they came to us and they said, in case of citrus rootstocks, they remain in the field for 24 months in the nursery, 24 months. That means 2 years. The reason is the stem should reach the pencil thickness when it is ready for grafting. Okay, it takes 24 months for it to reach pencil thickness. So they came to us and said, can you help us by through mycorrhizal inoculation, whether this period of 24 months can be brought down? So again, through a student's thesis work, to a fellowship was given to them by these people, and we did this work and inoculated is number one, and inoculated with various mycorrhizal fungi. You can see the best mycorrhizal fungus, which increase the stem diameter. And so in 18 months, the inoculated seedlings reached the pencil thickness compared to 24 months. Thereby, 6 months period, it can spend, it need not have to spend 24 months. So 6 months period in the nursery is reduced through inoculation. And now this is a very routine practice followed by the citriculturists in raising their seedlings. At this time, again, the floriculturists came to us. And we know that in Bangalore, in and around Bangalore, floriculture is a very, very important industry. And we know, if you know that one flight full of flowers go to Amsterdam. Earlier, it was almost going every day. Now, after COVID, it has come down. And see, that is the extent of export of flowers from our country to other European countries. So those people came and then asked me whether how will mycorrhiza help in flowering plants. So we did it with several flowering plants, but I'm just showing you one example of chrysanthemum, uninoculated versus inoculated. Definitely, you can see the number of flowers more here, but what we observed was the inoculated plants started flowering 3 weeks early. Okay, that is what the floriculturists were more interested. Okay, so they were said this is a very wonderful thing, and we have a post-harvest technology department in the university. So the post-harvest technology professor came and she said, can we take these flowers, both non-mycorrhizal and mycorrhizal, put it in the flower vase and see whether they last longer in the flower vase. I told, madam, you take and see what, it will be very interesting. And she came back and said, your mycorrhizal flower stayed 3 days more in the flower vase. I was really surprised. And now we have many reports with different flowers staying longer in flower vase from other countries. And of course, the reason was not known for a long time. Now we know in mycorrhizal plants, there is less production. The production of ethylene is less. And because ethylene induces flowering and also makes it to wither soon. But here in case of mycorrhizal plants, there is less production of ethylene that increases the vase life. Now we know the reason also why it is increasing. Again, Indian Council of Agricultural Research came to us and we have a problem in cashew. Can mycorrhiza help us? So another project came. And so the problem in cashew is the farmer, the nurseryman raises the cashew seedlings in poly bags, and the farmer buys the cashew seedlings and plants it in the field. And there is 40% mortality. That means if he plants 100 seedlings, 40 of them die. That is called as transplant shock. Okay. So the ICR wanted us to check whether the mycorrhiza can help in overcoming this mycorrhiza shock, transplant shock. And so that is how this was done in a particular research station, a cashew research station, which is very close to Mangalore, called as Ullal Cashew Research Station. Most of it was done in there. And you can see we developed one screen various mycorrhizal fungi and finally raised the seedlings and then planted in the field. And you can see the seedlings inoculated with these two fungi, Acylospora laevis and Blomus retinicatum. There was 100% survival when they were planted in the field. Whereas in the uninoculated ones, there are 63% survival. Again, a technology that is taken by the end user now. And this is, again, we are very happy that has been given to the farming community. At this time, the tissue culture people came and said how mycorrhiza can help in micropropagated plants. So we have inoculated this at the time of hardening. And in case of banana, you can see the difference with the inoculation. This is the type of difference. So, and this is another micropropagated plant, Ficus benjamina. The first slide is inoculated with the first pot is only inoculated with mycorrhizal fungus, and the last one inoculated with a consortium of microbes, mycorrhizal fungus plus Bacillus coagulans, which is a mycorrhiza helper bacterium, and Trichoderma harziana, which is a biocontrol agent, and also a plant growth producer. So you can see the difference between the two, which made us to think that inoculation with a consortium is better than inoculating only with a mycorrhizal fungus. So now we do the microbial consortium for us inoculant rather than using only mycorrhizal fungus. This is a very interesting plant because where we have a double road in Bangalore and the neighboring areas, this is the plant that is planted in the middle of the road because it can withstand pollution. So that's why Ficus benjamina is given so much of importance. And now we started working on microbial consortium, as I said. So this is in another forest tree species, Acacia auriculaformis, an inoculated plant, only mycorrhiza, microbial consortium. You can see the difference with the consortium. And this is a project which was funded by Department of DBT, and where we developed a consortium again. And in case of tomato, there is wilt disease complex. And again, it's very serious in Karnataka. Here, the seedlings and the plant are attacked by this disease, which we call as wilt disease complex, because it's not attacked by one pathogen, by several pathogens. And we have a fungal pathogen, and we have a bacterial pathogen, Ralstonia solani, and we also have a nematode pathogen, and Merodagai incarnata. A fungal pathogen, Fusarium, Ralstonia, bacterial pathogen, and Merodagai as a nematode pathogen. So that's why we call it as a complex. So through this particular DBT funded project, we developed this consortium consisting of a mycorrhizal fungus which takes care of the fungal pathogen, Pseudomonas fluorescens which takes care of the bacterial pathogen, and Phacillomyces lilacinus which takes care of the nematode pathogen. You can see the difference in the seedlings which the farmer can buy from the nurseryman and then plant it in the field. And we also tried it in the nursery bed because some farmers still try to raise their own seedlings in the nursery, though majority buy it from nurserymen. Even here you can see the difference between the uninoculated versus the inoculated. Another consortium which we developed for the aromatic crop plant, patchouli. Patchouli is a very important aromatic crop plant and it is known for its perfume, and it is used for making perfume, the scent. And so the student who was working with this particular plant, she used to harvest the plant and used to shade dry it before extracting the essential oil, and which is used by the perfume industry. And the whole laboratory, not the laboratory, the whole campus used to smell so good. People used to come and say, what you are doing in your lab. So it's such a wonderful flavor, perfume it has got. And now the Uttara Kannada district of Karnataka grows a lot of this particular plant because we have the industry for producing the perfume. And so there is a lot of scope that this particular technology of inoculation can go into that farming community. Another Here I wanted to show you the difference in the oil content which is used for the manufacture of perfume. In uninoculated control, the oil content was only 2.66, whereas with the inoculation with the microbial consortium, with 50% reduced nitrogen and phosphatic fertilizer, it was 3.66. This is what the perfume industry is very much interested. And another medicinal plant that was introduced in Andhra Pradesh, Tamil Nadu, and Karnataka is Coleus forskolai. And with the buyback arrangement with the pharmaceutical company. And farmers were very happy because there was a buyback arrangement by the industry. And they started raising the plant, but there was a severe disease problem, Fusarium wilt. And you know to what extent that damage? It is 85%. The disease index was 85%. This is a field experiment that was done in a place called Kollegal, which is in the border between Tamil Nadu and Karnataka. And here you can see 85% disease index. When we inoculated with only mycorrhizal fungus, the disease index was brought down to 68. But when we developed a consortium with the trichoderma, it was brought down to 33. So this is how a consortium is better than using only the mycorrhizal fungus. And of course, through another DBT funded project, we have developed the protocol for mass production of fungus, which has been taken by two industries in India, one industry in Malaysia and one in Indonesia. Inoculum placement, very simple experiments. I was wondering how people have not done it. Where to place the inoculum to get the best crop growth? And if it is a nursery raised crop, placing the inoculum 2 centimeters below gives the best result. If it's a poly bag, placing the inoculum 6 centimeters below the seed gives the best result. It's such very, very simple experiment, which is something new which we have to do and publish. And another thing is how much of inoculum is required for raising the nursery bed or in the field. About earlier we said 1 kg per meter square gives the best result, but we have come down off the level. 500 grams is enough for getting the best result. And here again, an interesting thing which I want to say about Krishi Mela, where we have to meet the farmers. Another Krishi Mela came, and the farmers were very happy what was expressed about mycorrhizal fungi and how they are useful in raising plants. And one farmer raised and asked a question. I have a 10-year-old mulberry garden. Can I inoculate? Is it possible to inoculate such old plants with mycorrhizal fungus? And will I be benefited by that? Will I get more leaf yield? Mulberry is grown for its leaf. So will I get more leaf yield? I told him, we have not done such experiments with grown-up plants. So you please come next year again. I will give you the answer. Fortunately, the sericulture department in the university had a 10-year-old mulberry garden. So we introduced the mycorrhizal fungus in the root zone of the mulberry of the plants, and then monitored it. And we could very clearly show that whatever was there, were the plants given 100% of phosphatic fertilizer, and the plants given 50% of phosphatic fertilizer with the mycorrhizal inoculation gave the same amount of leaf yield, or to some extent more yield than the 100% fertilizer. We did not stop there with mulberry alone. We extended with papaya. We also did it with clove with the help of the Spices Research Institute in Kerala, and we could see that inoculation of old plants in the root zone with mycorrhiza improves growth and yield of the plants. And it so happened the Planning Commission invited me as a special invitee to tell them about mycorrhiza. I was really surprised how they invited me. And again, the letter said, it is not, don't tell anything about science. What we want to know is about economics. So I had to prepare this particular slide to explain the planning commission members, which are mainly made up of bureaucrats. So this I prepared based on our results done on tomato. So the yield of the uninoculated tomato given 100% of phosphatic fertilizer would be 9,000 kg. And yield of tomato given 50% of phosphatic fertilizer inoculated with the selected mycorrhizal fungus is 10,400 kg. That means the farmer gets 1,400 kg increased yield of tomato. And you know that in Bangalore, the cost of tomato varies. Sometimes it comes to 2 rupees. It can go up to 80 rupees, sometimes 100 rupees also. So I have just taken 25 rupees on an average. So he is getting 1,400 rupees to kgs more, multiplied by 25 rupees, he gets 35,000 rupees more. And he has also saved 50% of phosphatic fertilizer. So add that to 35,000, he gets 36,875 increased income due to inoculation with mycorrhizal fungus. I think the IAS officers, the planning commission people, they were all satisfied with this economics what I presented, and they recommended that this should become a focus area of research, and funding agencies should fund research research projects in this particular area. And at the time of, it was a time that I had to retire from the university. And so I was also a committee member of DST, ICR, and so on. So one of the selection, one of the committee meetings, one of the DST person asked me the question, after retirement, what you are going to do? And I said, I will continue as emeritus scientist or professor in the university. Okay, you continue to do that, but don't stop your mycorrhizal work. You have to continue this work. So why don't you start a trust? When you start a trust, you can do all soil microbiology research in your work, in your trust. I thought it was a good suggestion. That is how we started this particular trust called Center for Natural Biological Research and Community Development, in short called as CNBRCD, where we are concentrating only on research. And we have the projects funded by various government agencies. And so the first project that came to me after we started the trust is this particular project being projected. And it had a very interesting story it had. One of the committee members said, all the wonderful research you have shown is the result of the work done by mainly your PhD students. And how many plants they have used? Maybe 100 uninoculated, 100 inoculated plants in the glasshouse and shown nice results. But if a forest nurseryman or a horticultural nurseryman raises thousands of seedlings, will your technology work? So they said, develop a project to see this. So that is how I developed this particular project. It was the first project that was operated through the trust. And again, there is a rule that a trust, when it is formed, they have to wait for 3 years to get a project funded by government agencies. They waived off that because I am already a committee member, the expert committee member. So that is how we started the project in the first year itself. So the 3 plants, this was on forest tree species, and we took 3 different tree species and 3,000 seedlings. I was scared how we are going to do it. One research fellow was given in the project, and this is the research fellow Raghu. Now he is associate professor of forestry and then the Karnataka Forest Department came to our rescue and said, do not worry. You want 30 laborers, 40 laborers, we will give you. But what we want to see is how the technology works. So this is how 3,000 seedlings were raised. This was all done in the Mandya district at 3 different locations in Mandya. And the forest department gave us all the support, and this is the result. You can see Tectona grandis, uninoculated versus inoculated. Dalbergia sissu, uninoculated versus inoculated. Acacia auriculaformis, uninoculated versus inoculated. So I felt that my students have done a good job, and whatever they have done works even on a very large scale. Even we take thousands of seedlings. And next thing we did was in in Mandya, some of you might have noticed, there is on the one side of the road, there is a degraded forest area. And you can see how degraded the soil looks like. And the forest department also helped us and said, why not we plant these uninoculated and inoculated seedlings in the degraded forest and see how they perform. And DST also wanted this because a DST funded project. So we again, it's a hill slope and it's very difficult to grow and plant. Again, the forest department gave a lot of labor to us and we planted it in this degraded forest. And you can see this is this acacia. The previous one was Tectona grandis. After 1 year of planting, uninoculated versus inoculated, this is in Acacia auriculaformis 1 year after planting. And here, this is very interesting. 52 months after planting, 52 months after planting, Dalbergia sissu, and this is the forest person who is standing the plants behind, and he looks big. And this is uninoculated. This is inoculated Dalbergia sissu behind him, and he has become small. The reason is the trees have grown much bigger. The inoculated trees were double the size of uninoculated plants. So the DST funded person, Dr. Vinita Sharma, visited Bangalore, and I took her to this forest, and she saw this and said, can your tiny fungus do this difference? I said, ma'am, you are seeing this. And she wants me to develop a project which goes to all the other parts of the country, and I do not have time. And to do this, I hope somebody will do it. And we also organize training programs for the forest nurserymen, horticulture nurserymen, and other people who are interested. And finally, what we can say that, again, yesterday I started with independence when there was shortage of food. Thanks to Green Revolution, high-yielding varieties were used. But what went along with that high-yielding varieties was heavy doses of fertilizers and pesticides. Now we know the deleterious effect, environmental pollution caused by the insecticides and the high levels of fertilizers. And now we are not only they are causing damage to the soil, animal, and human health. So this is the hot topic now of the United Nations, and the FAO is concentrating very much on maintaining sustainable agriculture or organic farming, where we can use less and less of chemicals like fertilizers and pesticides. And of course, you know that one of the states in India has already been developed as organic state, that is Sikkim. The Sikkim, you cannot use any chemicals, no fertilizer, no pesticides. So government of India is now trying to extend it to the other states. So it will come in future. So organic AMF, organic arbuscular mycorrhizal fungi, and other microbial inoculants play a very important role in sustainable agriculture. They are cost effective, much cheaper compared to fertilizers and pesticides. And more than that, they are environment friendly and they improve soil health. Again, soil health is one topic of current interest globally. Anything with the soil is healthy, then the plants growing on them are healthy, the animals feeding on them are healthy, and the human beings depending on plants and animals are healthy. So lot of importance is given to soil health. So these microbial inoculants, including mycorrhizal fungi, they are a boon to farmers, especially in developing countries, including our country, India. Thank you very much. And if you have any questions, I'll be happy to answer.
Thank you so much, sir, for this very insightful and interesting presentation. I am sure all the participants have a lot to take back from this session. Participants, if there are any questions, you can unmute and ask or put them in the chat box.